Hello, can everyone hear me? Yeah. All right. So as Cameron succinctly put it, I'm Kadin. I'm the current collections assistant for zoology. I was hired specifically for this cryogenics project, which I'm sure a lot of people have kind of heard about over the years, but have been kind of curious about. So I thought I would come up here and talk about it today. There we go. So for those who have not met me yet, I am originally from Minnesota, but I graduated from Colorado State University back in 2023 with a zoology degree. My background is a little bit in various types of museums, including CSU's own biology teaching collection, and I was here last year for an internship with zoology, working with their food collection. So I'm really happy I got to come back and work with this new kind of type of collection this year. I got some fun facts about myself here for those who are interested in that, but yeah, I travel a lot, been all across America, and I've been to various parts of the world. I love wolverines. I think they're really cool. And yeah, I've actually used to do archery competitively. So before we begin, I think there's some terminology I'll be using today that might be a bit unfamiliar for everybody, so I just wanted to kind of clarify those so there isn't as much confusion, hopefully. Cryogenics is technically the study of things that get really cold or how things interact with really cold things. We're going to be talking about cryogenic storage, so how we store materials at or below negative 80 degrees Celsius specifically. I'll be talking a lot about tissues, as that's what we store in cryogenic storage here at DMNS. That's just samples of organs and muscles. The ones that we most commonly collect here at DMNS are heart, liver, kidney, and muscle. We do collect other things when it is appropriate, but those are our main four that we use today. And you'll hear me talk a little bit about genomics, genetics today. Genomics is the study of all genetic code within an organism. That includes mitochondrial DNA and ribosomal DNA. I won't get too much into that today, but I will be mentioning it just to clarify what that is. All right, let's get into it. So this project really has started this year, as recently as July when it first opened, but really this kind of goes all the way back to the 1980s when DNA was first used in an analytical form. It kind of started out in criminology, but quickly started becoming something that we use a lot today in natural sciences, especially with the Human Genome Project that happened in 1999 in the early 2000s. It really became kind of like this data set everyone uses nowadays, which has prompted a lot of natural history collections to start collecting things specifically for DNA and genetic research use. These type of collections really need to be managed a lot differently than how we have historically. So skins and skeletons, those type of things don't really need this type of special preservation, whereas tissues need to be kept frozen or in ethanol or something like that. So in 2013, when we first started opening our new collections facilities here at DMNS, we started talking about different ways that we could store our current tissue collection, which all accumulated in 2024 with me being hired in July and opening up the Cryovault. So we use liquid nitrogen for our cryogenic storage. There's a lot of different ways you could do this, but liquid nitrogen is the one we chose for a couple different reasons. One of the big ones is less energy. As long as these tanks are closed, they will stay as cold as they are for probably a couple days, a little over a week maybe, which is a lot more than freezers can, as they need active cooling and a lot more active energy put into them. Another thing, liquid nitrogen gets very, very cold. Stays steady around negative 180 degrees Celsius. That's about negative 300 degrees Fahrenheit. So that is incredibly cold, way colder than a lot of our industrial freezers can even get, including our ultra colds, which only get down to negative 80 degrees Celsius. This helps a lot with DNA because it's a lot more stable at that temperature, especially without having to do a lot of more different like preservation methods. So that helps us have them viable for a lot longer than they would be in a negative 80 freezer. So where do you keep liquid nitrogen? It's not something you definitely want to keep in a cabinet or in the room where you work in your office. For those of you who have been here a lot longer than I have, you will probably remember where a lot of our collections used to be up on the second floor. We have something called the vault up there. That's where we used to keep a lot of our valuable specimens like gems and minerals. I think some of our anthropology was up there. Once that got emptied out, we converted to cryogenic storage specifically, so it got a lot more ventilation, has some more oxygen sensors, stuff like that. And we got 2 of these really cool tanks. These are 94K IC biomedical liquid nitrogen tanks. Sorry. And all of this was funded by the conservation fund that was set up a few years ago. So it's really cool to see that fund getting used for something like this and accomplishing one of its major goals of preservation of this collection and actually getting to see it come to fruition. Diving a little bit deeper into these tanks, they each have what are called dewars, which are essentially just containers, kind of like the same thing you keep gas containers in. They're called dewars after the guy who invented them. That was his last name. This is just so we can keep extra liquid nitrogen that we can feed into these massive tanks to keep them supplied over the course of a week. We do get these refilled every week, so if you ever heard a bunch of banging on the second floor, that was probably us moving them downstairs to get them filled. This is where we have this is where we have all of our tissues now, actually. We keep anywhere from 5 to 9 inches of liquid nitrogen at the very bottom here. There's a platform, and then we have 60 freezer racks, just like the one here. Each of these can hold 15 boxes, and within each of these boxes, we have these little tubes. Each of these have a single tissue in them, and there's anywhere from 81 to 100 tissues at each of these boxes. So that's where the 94K comes from. Technically, they can hold up to 94,000 tissues in each tank. So we have a lot more capacity than we used to, which is really great. Yeah, there's not much to them, honestly. We have a little control panel telling us a little bit of information about them, but these tanks just kind of chill, and that's great for us. So why am I getting paid? Why am I here? Most of my project has been transferring specimens. Before this project, they were kept in our vertebrate prep lab on the first floor here in ultra-cold freezers. So my job was to take every single little tissue and put them into a new box that was a little bit better fitted for these racks, barcode them or scan their barcodes, and then move them up into the cryo tank. So that took roughly 4.5 months to do. Actually just finished up that project. There's a lot more to this, including working with Arctos, which I'll talk about in a bit. But on top of that, there was a lot of maintaining these new storage tanks. They're pretty new, so there was a lot of learning curves for us. Sometimes they would yell at us and there would be alarms going off and be like, what's going on? So we had to kind of learn how to figure out what it means, what it's doing, making sure the temperature is always at the right time right temperature, getting the dewars filled, all that stuff. That's what takes up most of my day-to-day stuff nowadays. And since this, again, is all really new, I've written a bunch of protocols about what I'm doing, what can be done in the future, and kind of giving advice on what we should do in the long term to keep growing this collection. Oops. So we've been talking a lot about the tanks, but what actually are in these tanks? We have a little over 30,000 specimen tissue samples, at least, at the moment, which is a lot. The majority of them are mammals, at over 17,000 individual samples. The most sample that we have is the desert cottontail. If anyone's lived here in Colorado for any amount of time, you've definitely seen them. They're a little, you can see them in the parking lot, so they're pretty cute. I think one of my favorite tissues within the mammals, we have a field mouse, and its muscle sample is just its foot, which I think is kind of funny because it's just a little foot in a little vial. Our next most sampled is the birds. They're a little bit over 15,000 tissue samples. The most prominent of those is the horned lark. So those are also pretty common around here. And we have actually a lot of really interesting specimens between all of these collections that are kind of like personal to our curators or people who work there. For example, we have some Dunlin bird specimens that were collected by our current Zoology Collections Manager, Andy Dahl, in Alaska, which is really cool. And we got some stuff from their egg contents. And then we finally have herps. We don't currently have a lot of tissues from them just because we haven't really had the priority to work on herps, but now we're starting to work on them. So we'll see a lot of growth in here in the next few years. We only have about 81 of them right now. 26 of them are bull snakes, and we'll probably continue adding bull snakes to our collection. We get a lot of them from Denver International Airport. They do a lot of maintenance on their runways, and a lot of snakes, unfortunately, end up over there. One cool one that we kind of got fairly recently, actually. We got stomach contents from it, and apparently it had eaten so recently that we got 3 identifiable rodents, and 2 of them were able to be used as their own specimen. So that was kind of cool. Those are not frozen. Those are typical skin studies, but still kind of cool that ended up in there. But yeah, so this is kind of what's currently in our tanks. However, we will probably be growing and changing this over time. We may add other parts of our collections in here. These numbers will definitely go up over time. But yeah, that's kind of what's in there right now. And kind of going back to what I mentioned before with Arctos, not only are all these tissues physically moved into these new tanks, but they were all individually located into a type of hierarchy in Arctos, our current catalog for a lot of our zoology specimens. We basically created a virtual version of each box in each position. And with the barcode system that we started using a few years ago, we're able to kind of make sure everything was where it's supposed to be. So in theory, this makes it a lot more accessible, not only for us to utilize loan requests, but also just in general. This gives us a lot more information about what we have and where is it. So not only can people tell us that like, oh, we want these certain type of specimen, and be like, yep, we know exactly where those are. We know exactly what condition they're in, all that, which is super cool. This also kind of gave us a bit of an ability to double check some things, recover some tissues that were lost in between them being prepped and trying to get information from them. With 30,000 tissues, honestly, not a lot of them had any issues, so it's kind of good to find like those rare issues and kind of get them resolved. And so why we kind of do all of this, as I mentioned before, genomics and genetics have become really big in natural sciences. That really kind of makes these tissues not only a living collection because we add to them, but they're super important to the academic and scientific community. They may just start out as small samples from our specimen that we already prep, but once they get loan requests or kind of like general research questions, they usually go in for genomic testing or other type of research, which leads to a lot more scientific advancement and continues to kind of verify and really build this collection to be really important, not only for us, but also for the rest of our communities. And yeah, that's about it. If anyone has any questions, I'll be happy to try to answer them to the best of my ability. I also want to say thank you to all my mentors and my coworkers. This has been an awesome opportunity. And yeah, it's been a great time.
Lots of time for questions.
Hello. Hi. You said there's room in there for 94,000 specimens. Do you anticipate ever filling that up?
In theory, eventually, seeing as we're constantly getting new specimens and prepping them, we are hoping to possibly add some of our arachnology collection in there someday. Don't know what that will look like, but I doubt we'll reach that anytime soon, as of that is 94,000 per tank, and we have 2 of them, and we're maybe at quarter capacity for each of them right now, so maybe someday in like 15 years that'll be a problem, but yeah.
Thanks.
I can stop collecting pretty quick.
Yeah. I have a question. Of those tissues samples that you have, how many different species do you think are represented?
I did not count. There's definitely probably a few hundred per like group. So like mammals, birds, herps, there's only like a couple of them, obviously, because we have so little of them. But a lot of it ranges around like what kind of research was done previously or what we get donated. So we have quite a variety. Most of them are Western based or like stuff from here in Colorado.
And can you, I'm just going to hog it since I've got it. Can you also explain how, let's say a researcher wants to use a tissue sample. So can you explain that whole process of loaning and destructive sampling and whether we get it back?
Yeah, I can talk a little bit about that. I'm not entirely sure all the logistics because that's not my department. But most people will request what's called a destructive sampling or destructive sample that we don't expect to get back. So we only collect about a millimeter worth of tissue for each specimen, but that's more than enough because our current way of doing DNA analysis is really precise nowadays, so we don't need that much. So what we typically do, we grab all the tissues that they requested, we cut like the tiniest little sliver off of whatever they requested, put that into ethanol so it's stable for them when they get it, and we just put those tissues back into our collection to be, people can loan them again, and we'll just take a little bit off. There is some times where they need the whole thing for something else, and we will mention on Arctosis on loan, but we'll preserve that metadata and location for when it comes back.
Thanks. Other questions?
Can you talk a little bit more about the difference that this newer, colder storage method makes, and what was it like before? And obviously you had to be doing all the transferring from the old storage to the new storage. So I'm just kind of curious, if you could just expand a little bit about did you have to go sit in a cold freezer and do all these samples, move them all around? I'm just curious about kind of the context of all that.
Yeah, I can talk a little bit about that. So these are just kind of in a regular room. So the only thing that are actually cold are the tanks themselves. And you really don't want those open for very long because it is very cold in there. However, our tissues were stored in two ultra cold freezers in the vertebrate prep lab. They're very traditional type of freezers. So what you think about at home, just kind of more an industrial version of that. And those just having shelves. So I would pull out a box from one of those freezers. I would transfer it to a new box, or at least locate them all into their positions if the box was fine. And then I'd move it up into these tanks and locate it into a rack and all of that. That too was also just done in the prep lab at a normal room. I would create a temporary cold area with some coolers and stuff with a bunch of ice packs in it, just to keep them from thawing from over time, because it did take me a few minutes to kind of get them all done. But yeah, I was not sitting in a freezer on my own, just freezing. Yeah. Bit. Pick pass.
What's the shelf life of these specimens like? Are they good for a couple decades or hundreds of years, or yeah?
It's a little bit I was trying to look more into DNA preservation, and there's not a whole lot of talk about it, or at least not what I could find. I'm sure there's people who know a lot more than I do. Technically, everything degrades over time. That's just kind of a fact of life. But when they get really cold, that degradation really just slows down. And the colder it gets, in theory, the slower it gets. So these, as long as they're kept kind of in their containers, sterile and frozen, they can stay viable for, I'm sure, at least 100 years or so. I'm not sure what that real cap is, since DNA is pretty new in the grand scheme of things. So we haven't really been collecting it actively for very long either. Sorry. But really the major issue that people have with DNA preservation is the thawing and refreezing of it, because ice crystals can form and tear through it, and that causes degradation far more than just the natural aging of those tissues over time.
Thank you. So if that's the lifespan of the samples, what's the lifespan of the freezers? So you said that we might fill them up in 50 years. Are they going to be really functional? Could we replace a dewar rather than the whole tank? What's their lifespan look like?
So again, that's something we're definitely learning down the line. There's places who've had these a lot longer than we have, obviously, because these are very new. They aren't very active. Like I said, freezers who they need active energy input. They have motors. They have so much things going on in the background that if something breaks, it's kind of just down and out. Really, these are just like kind of glorified containers. There's not much actively going on besides like the monitoring systems and the pumping system to put more liquid nitrogen in. So, and those are all seeming like they seem to be fairly simple, fairly replaceable. So hopefully the idea is that these will be maintained for a very long time and hopefully shouldn't need a lot of maintenance or replacement. And the replacement really just be like, yeah, like another doer or something like that. So hopefully in the long term, this doesn't have a whole lot of maintenance costs either.
Other questions? All right. Give Caitlin a big thanks.